Hi everyone, my name is Christian Ieritano and I'm a PhD student in the Hopkins Group at the University of Waterloo. Today I'll be discussing the effects of field-induced heating that are pertinent to differential mobility spectrometry. But before we get into the fun stuff, it's important to understand the fundamental principles that underpin ion mobility spectrometry and why characterizing ion temperature is paramount for interpreting data from any IMS experiment. Whenever someone talks about IMS, the two main methods that immediately come to mind are either drift tube IMS or traveling wave IMS, since these two methods both either have a direct or empirical relationship with ion structure, which effectively allows us to peer into the gas phase structure of ions. Both methods work similarly in that ions traverse the mobility cell by being pushed by an electric field, while a drift gas opposes ion motion. The time that an ion takes to exit the mobility cell is correlated with its structure, as larger molecular species will exhibit longer drift times. The difference between the two systems stems from the operating pressure of the mobility cell and the nature of the electric field used, both of which can have a severe impact on experimental outcomes due to the possibility of ion activation and elongation. In drift tube IMS, ambient pressure conditions in a low electric field has little influence on ion structure. Drift tube IMS operates under the so-called low field limit, which effectively means that ion heating induced by the electric field, or thermal heating, is significantly less than the temperature associated with the ion's drift velocity. This means that in this regime, ion heating is usually insignificant, and the Mason-Champ relation between ion mobility and collision cross-section is valid. However, with the significantly lower pressures and higher electric fields used in TWIMs, ion heating becomes much more pronounced due to the inverse relationship with gas density. The effects of ion heating are especially noticeable for large flexible species and highlight the importance of characterizing ion temperature in order to rationalize IMS results. For example, a mobilogram of cytochrome C, a heme-based protein, was recorded in a SYNAPT G2 system at different traveling wave heights. At low wave heights, a distribution of compact conformations is highlighted in blue. As wave height increases, conformer populations shift in favor of a thermally unfolded variant that exhibits a greater collision cross-section. Quite obviously, as the wave height increases further, a greater degree of ion elongation is observed. So why bring all this up? Well, consider the operation of a DMS cell. Rather than a temporal resolution of ions, DMS functions by spatially separating ions based on their differential mobility under high and low electric fields. This is accomplished using a separation voltage, or SV, which consists of the combination of two sinusoidal waveforms. In contrast to drift tube and TWIMs, the electric field is applied perpendicular to ion motion. Effectively, ions are carried through the mobility cell with the drift gas, and adopt off-axis zigzag trajectories as they are subjected to the SV waveform. Ion trajectories are corrected by applying a static DC compensation voltage, or CV, allowing them to elute to the mass spectrometer portion of the instrument. The magnitude of this compensation voltage is drastically smaller than the separation voltage, so ion heating will originate almost exclusively from the SV. The duty cycle of this waveform, which is shown as a box waveform for ease of explanation, is split into 2/3 low field and 1/3 high fields. The amplitude of the waves are designed such that the integral over a single duty cycle is 0. For example, at a separation voltage of 3000 volts, the high field portion reaches a maximum of 2000 volts above 0, while the low field portion reaches a minimum of 1000 volts below 0. The nature of the waveform has important implications for ion heating that we'll be talking about shortly. Nevertheless, the point here is to show that the SV field strengths easily exceed 30 Townsends, which is well above the low field limit. Of course, ion heating in this regime is inevitable, and the effects can be seen for multiple chemical systems. In a select example, the Glish group looked at the internal energy deposited into peptides as a result of ion activation induced by the separation fields. In the case of bradykinin, the effects of ion heating increase with increasing field strength, resulting in the fragmentation of the +2 and +3 parent species, while a fragment ion grows in intensity. So I think I've impressed the importance of characterizing ion temperature, not just in the context of DMS, but for ion mobility in general. The work we've done to characterize ion heating was recently featured in the March issue of JASMIS, the methods and results of which will be the focus of the remainder of the talk. The models used to evaluate ion temperature are all provided in the publication in a ready-to-use format, and are freely available for folks to use and adapt to suit their own research needs. So to begin, characterizing ion effective temperatures requires some sort of chemical thermometer. To do this, we indirectly measure ion temperature by monitoring the degree of fragmentation of so-called thermometer ions, of which the benzylpyridinium species have been long used. Effectively, these species undergo a unimolecular dissociation that releases pyridine and a substituted benzyl cation. In the energy regime of the DMS cell, fragmentation of these species stops after this unimolecular decomposition, so evaluation of ion temperature doesn't become convoluted with competing fragmentation pathways. Depending on what R group is found in the para position, the bond dissociation energy of these species varies. By modeling the kinetics of the dissociation, the observed degree of fragmentation can be converted to an effective ion temperature. To do this, we take a page from RRKM theory, which is effectively the gold standard for describing kinetics of unimolecular dissociation. RRKM theory describes the rate of dissociation as a function of ion internal energy, E, and is essentially the sum of row vibrational states of the transition state that have an energy greater than the dissociation energy of the reaction, E0, divided by the density of row vibrational states of the ground state and weighted by Planck's constant. For unimolecular dissociations, modeling the transition state can be quite difficult. For benzylpyridinium species, it's convenient to treat the transition state in the phase space limit, which approximates the transition state as being equal to the separated products. This type of process is characterized by having no reverse activation barrier, which greatly simplifies enumerating the sum and density of states of both the ground and transition states. Doing so allows the relatively fast determination of the canonical dissociation rates for the 6 benzylpyridinium species used in this study. Now, since our RRKM rate is in terms of ion internal energy, we can weight it accordingly to bring it into the thermal regime. Consider that the DMS cell operates at slightly above atmospheric pressure, meaning that collisions between the ion and buffer gas occur roughly on the nanosecond timescale. Over a residence time of several milliseconds, this situation is a limiting case where there is rapid energy exchange compared to dissociation of the benzylpyridinium ion, and suggests that the ion's internal energy distribution takes the form of a Boltzmann distribution. The observed rate, then, is simply the RRKM rate weighted by a Boltzmann population at a particular temperature T. The Boltzmann population is simply the density of states of the ground state at the internal energy E, weighted by the population of that energy state at a given temperature. The Boltzmann distribution is then normalized by the partition function, which is effectively the sum over all energy states. Combining expressions and simplifying terms leaves us with the observed dissociation rate highlighted in blue, and is shown for the 6 benzylpyridinium species used in this study. Now that we have our dissociation rates, we can model the survival yield of each benzylpyridinium species by looking at their breakdown behavior. The probability of dissociation, which is effectively the fragmentation yield, is 1 minus the exponential decay of the parent species. Using a DMS residence time of 6.4 milliseconds, which is derived from the volume of the cell and the gas flow rate Q, and the fact that the survival yield is 1 minus the fragmentation yield, we can plot the survival yield as a function of ion temperature. These plots can be fit with special logistic functions to account for asymmetry in the sigmoid, yielding a tidy relationship that can be rearranged to solve for ion effective temperature given an experimentally obtained survival yield. Experimentally, evaluating the survival yield can be done by monitoring MRM transitions. We are essentially looking at 3 MRM transitions for each benzylpyridinium species, which correspond to the parent-parent, parent-fragment, and fragment-fragment MRM transitions. The fragmentation yield, and hence the survival yields, is calculated from the peak areas corresponding to each transition. Ion temperature at a specific separation voltage is determined by subbing in the experimental survival yields into the logistic fit function and rearranged to solve for effective temperature. It should be noted that all post-DMS potentials are minimized or set to 0 to ensure that ion activation and dissociation is occurring solely in the DMS cell and not in the mass spectrometer. With a method worked out, we thought it prudent to start our characterization with the most sensitive benzylpyridinium ion containing a methoxy substituent. This particular derivative has a resonance stabilized benzyl cation fragment rendering its dissociation energy much lower compared to the other derivatives. As indicated, the survival yield of about 0.65, in the absence of a separation voltage, significant ion activation occurs during the electrospray process. Following this black trace, in panel A, which corresponds to a DMS bath gas temperature of 373 Kelvin, it's pretty clear that changes in survival yield don't occur until a critical separation voltage is surpassed, in this case 3750 volts. Converting survival yields to ion temperatures in the regime where changes in survival yield are occurring doesn't present much of a challenge, but the question of what temperature the ion experiences before this critical point is reached is an important one. Looking back to panel A, as the temperature of the bath gas increases, the onset of dissociation of the species occurs at lower and lower separation voltages, as indicated by the red and blue traces. This in itself is an important observation, as it supports the hypothesis that ions are thermalized to the temperature of the bath gas after the electrospray process. This allows us to model ion temperatures before the onset of dissociation and after complete dissociation using two-temperature theory, which states that ion heating is the sum of the bath gas temperature plus field-induced heating. Two-temperature theory can effectively be simplified to a quadratic fit of the form y is A plus bx squared, where A is the temperature of the bath gas and B is a collection of constants related to the benzylpyridinium ion. These functional fits are shown as the transparent lines in panel B. Now, as a point of interest, in terms of correlating predicted effective temperatures with those determined experimentally, the nature of the asymmetric SV waveform must be taken into consideration. The maximum voltage under the high field condition is 2/3 of the peak-to-peak voltage applied. So again, for example, at 3000 volts separation voltage, the max potential is actually 2000 volts. In terms of ion effective temperature, ion heating is going to be at a maximum under the high field condition, which therefore defines the fragmentation behavior of the ion for each SV cycle. Since the ions undergo approximately 20,000 SV duty cycles during their transit through the DMS cell, the observed effective temperature must then reflect the sum of the bath gas temperature and the heating imparted during the high field portion of the waveform. This analysis can of course be extended to the other benzylpyridinium derivatives, but due to their higher dissociation energies, SV-induced fragmentation is only observed when a bath gas temperature of 450 Kelvin is used. Using a similar two-temperature theory approach, ion temperatures can be modeled before the onset of dissociation for each of the remaining benzylpyridinium species. At the maximum bath gas temperature and separation voltage our instrument can reach, ion effective temperatures can reach up to 759 Kelvin. Under the same activation energy conditions, there is clearly a relationship between the observed ion temperature and its structure, which could possibly lead to a way to model ion temperatures as a function of its internal energy distribution. So to look at this, we need to consider the activation conditions within the DMS cell and how internal energy is deposited into the benzylpyridinium species. The total energy imparted to any ion is related to its collision frequency with the buffer gas, which is proportional to its collision cross-section. By this logic, larger molecules traveling at the same velocity as smaller molecules will gain more internal energy. However, since geometrically larger species have lower mobilities in a given collision environment, larger molecules will travel more slowly than smaller molecules of equal mass, and will hence exhibit a lower kinetic energy. In other words, ion internal energies in the DMS are governed by two opposing effects. Ion internal energies will increase with ion collision cross-section, but ion temperatures will also diminish as collision cross-section increases. This can be rationalized by thinking about how the molecule partitions its internal energy. Once collisional energy has been deposited into the molecule, that energy gets distributed through its various row vibrational states. To a first approximation, the density of states of a molecule can be described by its mass-weighted degrees of freedom. For example, the collision cross-section of the 4-fluoro and 4-chloro benzylpyridinium derivatives is similar, and their number of degrees of freedom is identical. However, the fluoro species has an effective temperature that's nearly 20 degrees hotter than the chloro. This stems from the lower energy row vibrational states accessible to the chloro species thanks to its heavier mass. Treating the benzylpyridinium suite as a whole, it's easy to ascertain relationships in ion heating using the relationships of collision frequency and how a molecule partitions its internal energy. Plotting ion effective temperature as a function of the mass-weighted degrees of freedom and inverse collision cross-section yields linear relationships at every separation voltage sampled, effectively providing an avenue to predict ion effective temperatures using only intrinsic ion properties. This linear relationship of ion temperature with its internal energy structure begs the question of whether first principles methods can be used to model ion temperatures in the DMS. Effectively, one can make a self-consistent evaluation approach using only the Mason-Champ relationship with collision cross-sections modeled at various ion temperatures. Initial evaluation of the ion mobility K at the temperature equal to the bath gas temperature allows for an initial guess of ion effective temperature using two-temperature theory. This new effective temperature is then used to re-evaluate K using the Mason-Champ relation, which is then subbed back into the two-temperature theory expression to obtain a new effective temperature at that particular field strength. This process is completed iteratively until effective temperatures agree between iterations to 10 to the negative 4 Kelvin. It should be noted that due to the asymmetric nature of the waveform and the fact that each benzylpyridinium ion will undergo over 20,000 SV duty cycles, the effective temperature observed will reflect the amplitude of the high field portion of the waveform, as I mentioned previously. Comparing temperatures predicted by two-temperature theory to those determined experimentally makes for some interesting observations. First and foremost, the agreement between the two is excellent considering the simplicity of the model, where mean deviations are shown for the methoxy derivative, which agrees best, and for the chloro derivative, which showed the greatest deviation. Two-temperature theory has long been the primary method to assess the impact of ion heating, so agreement between model and experiment isn't too staggering at low ion temperatures. However, it's pretty obvious to see that deviations become much more prominent as ion temperature increases. This stems from the limitations associated with two-temperature theory, which become increasingly relevant as field strength increases. To talk about these briefly, the model uses the Mason-Champ relation, which breaks down above the low field limit due to the dependence of ion mobility on electric field strength. Moreover, the waveform is actually bisinusoidal, so the instantaneous ion velocities induced by the electric field will always be less than the traditional description of ion velocity. Simply put, ions do not instantaneously adjust to the dynamic field, leading to this inequality, which again will be more pronounced as field strength increases. Finally, at sufficiently high field strengths, it's possible that additional heating can occur during the low field portion of the waveform. There are, of course, other factors that influence the degree of heating imparted to ions by the separation voltage that couldn't be covered due to time constraints. But in summary, what we know is that once a critical field strength is surpassed, significant fragmentation of the benzylpyridinium ions is detected. At the maximum bath gas temperature and separation voltage that our instrument can accommodate, effective temperatures for the benzylpyridinium species goes as high as 759 Kelvin. The extent of activation at a given separation voltage correlates linearly with ion structure, as well as with predictions made by two-temperature theory. But the real bread and butter of fundamental research is how it can be applied. Currently, our work in determining ion temperatures has been a staple of our drug discovery program, notably in the prediction of physicochemical properties. Effective temperatures are used to assess the dynamic conformations that molecules adopt throughout their transit in a modified DMS environment, which serve as the partial input for ML model training. This work also has applications in evaluating reproducible SV-CV pairs for different ionic species. We are working on an SOP that uses benzylpyridinium ions to assess the performance of the SV waveform using a combination of SV-CV pairs in the form of a dispersion plot, as well as monitoring survival yield to assess the consistent performance of the SV waveform. And with that, I'll end things off by acknowledging my supervisor Scott Hopkins and the remainder of my PhD committee, especially Professors McMahon and Dr. Campbell for their collective insights and support. Of course, an equal amount of gratitude goes out to my lab mates, and I'd also like to acknowledge our various funding sources. For any further questions, we invite you to read our recent publication in JASMUS. Otherwise, I'd be happy to address any concerns during the ASMS watch party. Thank you all for your attention.